But no, thanks, buddy. Okay. Well, hey, everybody. My name is Matt Huber, and this is Algae Research and Supplies, one of our lectures on growing microalgae. I have my lovely son and assistant, Finn, who's going to be joining us. Right now, he's getting ready.
Buttoning up my shirt.
Here, just look up. This is Finn. He's going to be helping us out today. But for right now, won't you stand over there? You can be just off camera. Okay, today we're going to go over a handful of things, and Finn, you're going to kind of be my the human I'm going to be teaching to. Okay, so why don't you come right over here, Finn? I'm going to put this to the side, and we're going to be going over these things today. Okay, my son Finn is going to be going into the 6th grade this year, so this is one of our opportunities to be teaching is in the middle school market. Okay, so today we're going to do quick introductions, which is what we're doing right now. My name is Matt. This is Finn.
Hello.
And then what we're going to go into is we're going to do some comparisons between plants and animals. And those comparisons are going to include their anatomy, the environment with which they live, how they reproduce, and then we're going to get into some culturing methods. And then we're going to talk about one of our culturing kits, which is a really simple one, which is for growing the microalgae Nannochloropsis. Okay, so let's go ahead and start. I'm going to do quick is this right here, a little bit more light on us. Okay, so comparison between plants and animals. Then what's the difference between plants and algae? What's a plant?
A plant is.
Well, no, just tell me what a plant is. Go off the top of your head.
A plant is a living being that makes its own sugar to survive from the light.
It does.
And then algae is a microorganism that kind of does the same thing.
Yeah. Well, algae doesn't need to be a microorganism. It can be a macroorganism too. But you hit the nail on the head. What do they have in common? Plants and
they both make their energy from the sun. From
the sun, or from just light in general. Because we can grow algae using artificial light, can't we?
Yes.
Yeah, that's very good.
Such
as these things. We've got these big light tubes right there. So let's talk a little bit about what the differences are between algae and plants. So let's look at their anatomy first. Finn, what's this?
This is a flower.
Some kind of a flower, that's right.
It looks like a poppy because of the orange.
Yep, that looks pretty similar to that. So this is a flower. Where does the flower get its, or where does this plant get its energy?
It gets its energy from the leaves, through photosynthesis. It creates sugar,
which
inevitably gives the plant ability to reproduce and energy to grow.
It moves the sugar up and down through a system of tubes, and that sugar is dissolved in what?
Water.
It's dissolved in water, and it moves around using water.
That's kind of what nectar is.
That's exactly what nectar is. Nectar is sugar water. So we have the photosynthetic tool that the plants use is what? The leaf.
The leaf.
Where do they get their water from?
They get their water from underground water sources or
through
rain,
through their root system, right?
And they gather that through the root system.
They gather that through the roots. So they rely on water a lot, and they get the water into the roots, and that comes up through the stem and goes out to the leaves. Okay, that's really good. Now, in microalgae, tell me how they get their water. How do they acquire water?
Through their skin.
Yeah, they absorb it right through
like
their cell membrane,
like frogs and toads.
Like frogs and toads, exactly like that. It passes right through there, but they don't have skin per se. Algae don't.
They don't have.
They just have a cell membrane, right? Yeah. So that's something so the plants and algae, do they have that in common where they need water?
Yes.
Yes, they absolutely do.
They both need light and they both need water to create energy to survive.
Okay, so where do plants get their nutrients from?
The sugar that they create.
They get their, but they create the sugar give me your off camera a little bit. They create the sugar, but through photosynthesis, but they get their nutrients from
The soil.
From the soil. The water goes into the soil and it dissolves some of the, no, sorry, honey. They dissolve the soil, they dissolve the nutrients in the soil, and the roots take those in and they bring them up into the plant. So where do the nutrients come in with in algae?
The nutrients come in.
They come in the water, with water.
In the water.
They're just dissolved in the water, and they go across the membrane, and they're absorbed into the plant or into the microalgae itself. So, tell me a little bit about how microalgae move. Do you know how they can move around in the water? You might not know this.
There's currents. I think.
Do you know what this is?
They're filbum.
It's called a flagella. So microalgae can actually move. Some of them can, and they use these little things called microtubules. Or they have 9 plus 2 microtubules. They're flagella, and depending on whether they're eukaryotic or prokaryotic, they have different types of flagella. And these flagella, they move like this, and they act.
Tail.
They do. They act just like a fish tail.
Like our goldfish.
Yeah, we have our goldfish over here.
Yeah, we actually like huge, like this big.
They are pretty big, and this allows them to move. So, what do you think they want to move? Why do you think microalgae might want to move?
Say on probably like the day we're having right now, there could be clouds and there could be sun.
Where would they want to go?
And then they'd want to move to where they could get the most energy. And it's for sunbeam or rays.
That's exactly right, Finn. They want to
So
move
they
towards the
wouldn't
light.
want to be under the clouds because the clouds are blocking the sun.
Well, in water, if you swim, have you ever swum down in the ocean or in a lake? What happens if you swim down? Does it get lighter or darker? It gets darker. So these plants, these microalgae, they've developed ways to move around in the water column to get light. What other things do you think they might want to swim towards?
Other food sources?
Say nutrients. Oh, other food sources, that's right.
Yeah, like some algae eat algae.
Some algae eat algae, they do. But a lot of times
really algae.
They do. So one of the things that they want to do is they want to swim towards nutrients. And they can sense on their bodies whether there's a higher or lower concentration of nutrients, and they can make their way there. It's kind of cool. They kind of like sniffers, like Moxie, our dog.
Like in Godzilla, in King of Monsters, like they use the radioactive trackers to like track Godzilla.
They do. So that's like a radioactivity tracker for Godzilla. Right.
Except this, they can tell there's nutrients in the water.
Yeah.
But for Godzilla, they test there's nuclear energy in the water.
Exactly. So tell me, how do plants move around? If microalgae use flagella, how do plants move?
They don't.
They don't move around.
The seed is planted in the ground, but once the seed grows, it can't exactly move unless it's moved
Right.
or acted on by another force.
There you are. So we could transplant it, but we can't get it to move itself.
Sunflowers will angle themselves to catch the most sunlight.
That's true. So we do have what's called thigmoplasty, which is when a plant will go and move its flower head or its leaves to best get itself towards the sun. That's called heliotropism, or helio means light and trope means to move, I believe. So that's heliotropism.
And some plants, I think some species of vines, if they're in shade, they will spread out until they find the sun, and then they'll start to grow more and more leaves in the sun.
Excellent. I just want to make sure that everybody who's allowed in, can you check to see if there's anybody waiting in the queue? I don't know if there's a queue or not. 1 second while we allow everybody in. Share screen, manage participants. Are they in? One person is waiting. Admit. There we go. Okay, cool. If you can monitor that, that'd be cool. Yeah. I don't know if there's a way to do that. Okay, so we've now, we just went over some of the comparisons of plants to algae. We've gone over anatomy, and we just went over motility, or mobility. Another way to say mobility is motility, because you're motile if you're able to move. Next, let's talk about their environment. Tell me, Finn, my foil, where do plants live?
Plants live Almost everywhere except Antarctica and the North Pole.
They're all over, and are they on water or land?
They can be on both.
But they're generally on.
They're generally on land.
And what do they live on? I'm getting to this. What do they live on?
They live off of soil and ground.
Yeah, they live on what we call terra firma, or just soil, ground. They can live in any kind of environment there. But they live in dirt, and they have the roots that come in contact with the ground. And that's a good thing to remember when we're talking about where they live. So this is kind of their environment. What other parts of the environment do plants touch? They touch the dirt, and they also touch the
air.
They touch the air. What's in the air that plants use?
CO2.
They use CO2. That's such a good answer.
They use CO2, which is what we breathe out, and then they put out oxygen, which is what we breathe in.
My goodness, oxygen is a waste product for plants. Do you believe that? It's almost like plants and animals are two sides of the same coin, where we consume oxygen and we exhale CO2, and plants breathe in CO2 and they exhale oxygen. It's kind of a cool concept right there. So that's on land where plants, terrestrial plants, have they have their roots in the soil and they contact the atmosphere.
And that's also kind of what happens in the seaweed and in lakes, lily pads.
Yeah, so
lily pads take the soil from the bottom, or seaweed as well, and then they get nutrients, and then they get the sun when it comes in. That's why you can see them floating on top or closer to the surface. And then they also get oxygen from the water.
That's right. Well, let's get to that in a second. So what Finn's trying to say is there are some terrestrial plants, some plants that live in shallow water, and they can put their roots down into the ground. But here's the thing now, let's talk about microalgae, not macroalgae, but just microalgae. Where do microalgae live? Do they touch the soil?
They would if they wanted to.
Not necessarily. Do they are
They don't need it, but
they are
could.
suspended in water. Matter of fact, we have some Nannochloropsis, which is a green algae right here. These are very small, and they stay suspended in the water column their entire lives. So are they touching soil?
No.
Are they touching the atmosphere?
Yeah.
Ooh, what do you mean?
There is a slight bit of atmosphere in the water. That's how fish breathe. They use their gills to take in.
They do. How does that water? So what happens there? There's no atmosphere. The gas that we know as our atmosphere is at equilibrium with water if there's enough surface area. So the gases, the oxygen concentration is the same in the air as it is in the water. And also what gas?
CO2.
The CO2 is at equilibrium as well. So these plants can go ahead and suck in the CO2 and they give out O2. So that's their environment. So the water environment is a little bit different than the plant environment, right? Than the terrestrial plant environment. Quite a bit different. So let's talk really quickly about how plants might reproduce. How do plants reproduce? Do you know?
Let's take bees as an example.
Bees are pollinators, and those are only for what we call right, which are angiosperms. And these flowering plants, the bee helps to pollinate the plant, and it makes a seed, and then the seed goes and disperses and can spread the plant around. Now, do you know how algae do it? Do you know how algae reproduce?
They just pop into thin air.
You know what? They don't pop into thin air, but they do something that's similar to that, and it's called binary fission. How many are in a bicycle? How many wheels? 2 wheels in a bicycle. How many different parts do you think are in binary fission? 2. Very good. They go through and they will actually separate the cell in 2 smaller parts and then separate. Fission meaning to separate, and binary 2. So
and those cells will
separate and grow as well.
Divide as well, but not before they usually get a slight bit bigger.
Yes, and then they'll grow, and as they grow,
they'll separate.
Then once they get large enough and they decide to. Now, plants go and they do sexual reproduction using flowers. Some, the angiosperms do. Others do as well with different methods. But there are some, what happens with algae cells, which is kind of interesting, is they can go through hundreds of reproductions, and then when the environment changes, then only then might they say, okay, we're going to form a different kind of cell, a resting spore or a reproductive cell, and they change the whole shape of the cell changes, and then it can go through, some of them actually go through phases of sexual reproduction where they actually will separate into haploid cells, but that's going to get for the high schoolers a little bit later on. So that is now, we've covered anatomy, we did some on mobility, we talked about the environment that they live in, and a little bit about reproduction. I want to ask you one question real quick, Finn. Do you know what the major nutrients are that make up our bodies? What are the major chemicals that we take in? You said one of them a minute ago with the exhaled gas.
We put out CO2, we take in oxygen.
I'm going to stop you right there. Do you know what the major nutrient or the major element is in protein? Anybody? Danielle?
Helium.
No?
Helium.
It's not helium. Our helium quotient as a mammal is very small. The major thing that we want to use as mammals is nitrogen. So we take in nitrogen in our protein, and then when we urinate, we excrete nitrogenous waste. So this is a chemical that we can add to our media, which we could call soil your media to grow plants. Or you could call the water, the liquid that we grow microalgae and media as well. And this is sodium nitrate. And this is one of our macronutrients. We call them macro because they're a big part of the nutrient media. This is a macronutrient for there. So when we go, is it possible for you to grab one of the fertilizers from the corner over there, the plant ones? When we talk about growing plants, we talk about NPK, nitrogen. What do you think the P stands for? Say phosphorus. And then the K stands for, do you remember K from the periodic table? Even though it says K, it's really a P. It's potassium, NPK. So on pretty much every type of nutrient or fertilizer you'd use for your plants, there is going to be on the back, oh, here we go. There's a guaranteed analysis of NPK, which you can see right there. This is a 343. So what most algae want is a 1011. So that's why we created our own basically 1011. It's a derivative of F/2 media. So we have we're able to put in nitrogen. You ready? NP. This is potassium. And that's our NP. And I guess I didn't pull out the phosphate. But there are also other things too. Let's pull this whole tray over here. I do, because this is kind of the analogy of it here. These are kids' vitamins, right? But for when we're talking about making media, for when we're going to make our own nutrients for the microalgae, we're talking about nitrogen, potassium, and phosphorus, which I don't think I have out here. But there's also things called, those are the macronutrients, and then we're going to talk about the micronutrients, which include the big micronutrient that's very important, is this sticky stuff here. That's iron. You got to have iron. It's a key part of photosynthesis because that iron molecule is right in the middle of the chlorophyll molecule. And without that, you can't go and do photosynthesis. You have to have iron. See, we have zinc as well, and we also have crazy small amounts of cobalt. It is really poisonous, but it's necessary in small amounts. And then also, I've got some calcium chloride here. Calcium we use in our bodies. Yeah, our teeth are made. And what else is made of calcium?
Fingernails.
I'm sure there'd be some of them in there. But what about inside here?
Oh yeah, bones.
Your bones. Now, microalgae use calcium as an ion that goes in and out of the cells, just like we do. We have calcium ion channels and they help to give the cells charge. So they'll pump out a whole bunch of calcium with this positive charge, and then they can use that literally an electric charge. It's a zeta potential to do stuff, and they can either try to get other ions inside their cells. It's kind of a way to basically charge them like a battery. So this is another one called manganese, which again you don't want to eat it, but it's super important in very, very small amounts for enzymatic actions. Okay.
Yeah, like you wouldn't want to eat gold, but we do have some in our bodies.
I don't know if gold is an essential nutrient. I think it might just pass right through us, but it's a good observation. I know that there's a product called Goldschlager, which was very popular back in my youth, which may or may not be important as part of a complete diet. Probably not. So, let's see. So we've covered a whole bunch of different things about the nutrients. We've talked about where algae live, how they move around, the environment that they live in. So now let's get into some culturing methods, okay? So what do you think algae need, Ben, just from knowing about where they live in the wild?
They need water.
So they need water, okay? And I'm going to get some of these things out while you tell me, okay?
They need water.
Here's water.
They need light.
They need light, and we've got some light stuff here. Why don't you go on in there? They need, oh, it's okay. Come on over here.
They also they need the nutrients.
So they need some nutrients, and this this is an example of some nutrients here. So, and then they also need
something to make them reproduce.
No, well, what are these?
Seawater, culture salts.
These are the salts. So we need to make their environment for them. We need to make their environment so that it's healthy for them. This example here is Nannochloropsis, and this is a seawater organism. So we want to make seawater down. So we bag up different kinds of salts that will mimic a good seawater, and then we can add that to drinking water. This one is actually made, this package is made down for 1 gallon. So if you were a teacher and you wanted to have your make 1 gallon of salts, and that way you could pour it into a whole bunch of different students' stuff. You just want to get this 1 gallon pack, put it into a bottle of drinking water. Spring water is best. Why spring water? Because it has also a handful of those micronutrients that the microalgae will need. So then you could take this down, add our nutrient mixture to it, and then that's now your media. Okay, what's that? Okay, cool, thanks. So that's the media right there. So once you're culturing, you can put that in any container you like. Now here in the lab, I have a whole bunch of these things. Do you know what these are called, Sid?
That is called
Oh, if you get this, I'm gonna totally give you a high five. Starts with an E. 3, 2, 1. Danielle, what is this?
Erlenmeyer Flask.
Say it really loud.
Erlenmeyer Flask.
This is an Erlenmeyer Flask. That's right.
That's a flask.
This is an Erlenmeyer Flask. What is this thing called? Do you know what these are called? This container?
Oh, I've heard all these before.
Starts with a B.
Beaker.
It's a beaker. That's right. All right. So here's another one. This is a larger beaker, or larger Erlenmeyer Flask. This one is a culture of Isochrysis, which is a haptophyte. And we use these a lot. We culture this a lot for the aquaculture industry. And a lot of times what we'll do with situations, or when we're trying to grow in here, we'll add the seawater media, and then we'll add what's called an inoculum. So that would be another amount of algae in there to start it out. It's kind of like the seeds. So, and we're going to do a demo here in a minute. We get a half a liter bottle of water. So we'll show you a demo of that in just a second. But another way you can do it, you don't need to use necessarily.
So what you're saying is when you put the.
So another container you can use if you don't want to use an Erlenmeyer or a beaker, you can use a giant carboy like this. Or, hold on one second. Or you could, if you have a 10-gallon aquarium in your classroom, you can use those as well. So, and those are other really good ones to use. This is Flagella vulgaris. So this is one of our workhorses here in the lab.
Yeah. And what you mean by it's the stuff again? What you mean by when you use this to sell to the big industries?
Aquaculture.
An example for like the starter would be if you have a citrus tree that you just got, you could add the citrus like fertilizer that I think we had over there.
Yeah.
That would be like the booster like they have in the algae.
So nutrients. Okay, cool. So to give you an idea too, not only could you grow algae in these small containers, but I've worked at big farms that are bigger than this one here. This is a production facility scale farm for microalgae. And they can, these farms, these big ponds can get to be up to several acres large. The biggest ones that I've seen have been up to 5,000 square meters, which is a little bit over 1 acre per pond. And there's a whole bunch of those. And this is how when you go to the store to buy spirulina in a tablet form, they grow them in farms like that. But real quick, let's go ahead and do a setup here with one of our kits. So this is the Nannochloropsis kit. You want to do this for me, Finn?
Sure.
Yeah, take those things out. Show them what they are really quick.
So we have the tube. I'm assuming that's going to be
tissue. That's called a tissue culture flask.
Tissue culture flask.
Yep.
We have the culture salts, and then we have the starter nutrients.
Those are the nutrients, exactly. So what we do is you add the salts.
Then we have the instruction manual if you need that.
Yep, and there's a whole manual inside there. Very good. So will you add the salts right to this, please? Into this container. The salts are all standardized to go into a 500 milliliter bottle of, it's best again with spring water, but you can use drinking water as they have it there. You want to add the salts to it, just like that. And then Finn's going to demonstrate adding the nutrients to it as well. You got it, Bucko. We glued that one on for you.
Oh. So I'm getting special treatment.
Yes. Yep. Put it all in there. Yep, you got it all in there. So now this water has now become, you kept that for us? Cap it and screw it down tight, has now become algae culture media. This is now perfect soil, but for your algae. Now shake it up to dissolve the salts. Yep, turn it upside down. Get it. While he shakes that up, I'm going to be your helper here. Now, will you add those? This is the inoculum. We usually put 2 of these in just so that if you drop one, you still have some more to use. And that's going to go into your tissue culture flask. Yes. Well, you might need to just tap it. Tap the end. There you go. Very good. One.
And here's 2.
You just got the other one going right there. Perfect. Okay, there you are. Nice.
And now
these are almost completely dissolved, so I'm going to give this to you, Finn, and you can.
It's like a tornado. I always love it when water does that.
All right, pour that in there, and you're only going to pour it up to the first shoulder there because you want to have a lot of space in between the. You want to have a lot of surface area. Let me get it there. Perfect. Now you can tap this and put this in your refrigerator or just somewhere that's in the dark, just in case there's a few algae cells from the air or there's in there. Now this is a perfect place to go grow algae, so put it somewhere in the dark. Then tap that, and
this will start growing.
Yep, so you can set that in a bright place. You don't like direct sunlight because that's going to get too hot. If the algae cells get to be over 100 degrees Fahrenheit, they could begin to die. So we want to have it somewhere warm and bright, but not too bright. That's Nannochloropsis. So every day you want to shake. Every day you want to shake it. There's something here. Every day you want to shake it. Will you shake it, Finn? No. Will you shake it? I made it.
Okay.
Okay. So, and that is, and this is.
It should look like this.
Yes. This is going to get to be this dense.
Yeah.
If you pick it up and shake it every day, this will grow into that. And one of the neat things that we have too is we have the ability to quantify how much algae, how dense the algae is growing. Now this is a separate unit, but this is called a, what is this?
Fission stick.
This is a Fission stick, exactly. So as you
use to measure the density of something.
Exactly.
Do
you mind if I do it? We're running close on time, so I'll be really quick here. So the Fission stick uses what's called optical density. That little target right there on the stick. You want to take this looking straight down at your culture, and you want to push the stick underwater until you cannot see the target anymore, until you can't resolve that target. Now you want to remember how, as you do this, you want to remember what that target looks like when it disappears, and then note how deep under the water the stick measured. So in this case, this gets to be about 75 millimeters. Do this one here.
this one.
And they're written right there. So as your culture grows, you can use the Fission stick to monitor and quantify how much algae culture is there. Now the neat thing
This
to do
one is 45 inches.
45
millimeters.
Millimeters. So what you can do with this is you can start tracking your biomass over time, or the Fission stick depth over time.
This one's about 30.
So your graph is going to look like this at first, where it's going to be your Fission stick depth is going to be dropping off, but that's because the biomass is getting bigger. Now you can just invert that to have the shorter the Fission depth is the most biomass. So if you just turn that upside down, you can have it actually.
This one is very dense. This one is 20 millimeters deep before you can no longer see the color.
Very good. So that's pretty much our little demo for today. I'm going to go ahead and say thank you guys. We're going to open it up to questions in just a minute. So if you guys have your, I'm going to turn the microphones on. You have questions for Finn or myself, we're happy to take them from you.
It's also my birthday today, so.
Okay, let's see here. Mute security, manage participants. No, I don't think. If anybody has questions, they can go ahead and shout them out. The top of my head is easy to see there. So.
All right, I've got a few questions, but I'll just start off with one. When for a thing of spirulina, if you harvest out some of the spirulina and then you're filling it back in, do you want to have the medium and the salts in there, or just the food part? Because that and I guess also why, because I would think maybe the salts wouldn't get consumed by the
that's correct.
Algae.
Conserved, but the nutrients are not. So what you'll want to do is you'll want to put more of the we call it the F/2, or in your case with the spirulina, it's the spirulina nutrients. You'll want to add those back in. So for every liter that you harvest, we recommend adding a milliliter of the nutrient back in. Okay.
Okay.
Now with that, we don't know how dense it was, so that's a good guess as to where you're going to go. So two things. One, you should monitor your nitrogen. Nitrogen's a good indicator for the whole system of consumable nutrients that are going to be exported as you harvest the spirulina. So with that, you want to make sure that you just use a nitrogen test strip to figure that out. And also, outside of the nitrate, you want to do something called blowdown. And that is for every liter that you harvest, you kind of want to get rid of about 100 milliliters or 10% of your harvested material and just throw that out and replace it with 10%, that same amount, of brand new media. And what that's going to do is it's going to limit, it's going to prevent something from building up If you have a closed system, you might get just some analytes, whether it's a crazy, wacky nutrient or a waste product that the algae put out, building up over time. So if you look up on Wikipedia for blowdown of a water heater, it's exactly the same concept there.
Okay.
Any other questions?
I have a question. Hello, how are you?
Hi there, how are you?
Thank you very much. Because I arrived very late, I don't know how you start, sorry. I want to ask about only you work on spirulina, or do you have another species for culturing?
Oh, we have all sorts of species. So just the top level, we go from freshwater. Spirulina it's a bit of an extremophile of the freshwater. They like really alkali water. And then we go all the way through brackish into seawater. So there's strains across the board that we culture and sell here in the lab. So lots of different organisms from the ones that are in front of us. This is Nannochloropsis. You saw earlier. We've got Isochrysis. We've got Tetraselmis. We've got we've experimented with Pyrocystis fusiformis. That one's a bit of a pain sometimes, and so we're not doing that one. But that's the bioluminescent one right now. Yeah. Another
question I have: how we can isolate this pure microalgae from the seawater? I have problem about the isolation. This is very difficult for me. How I can isolate and again culture by pure culture.
Okay, so you're looking at isolating. You're looking at finding your own cells in seawater and isolating them.
Yes.
Okay, that's a great question because I had to do that in grad school quite a bit. So what you want to do there is go get your sample from the water. Okay, and there's a couple different methods you can use. One that I like, that I think is the easiest, is the dilution. They all require patience, first of all, a lot of time. So you'll want to take your sample, and then you'll want to just dilute it into filtered, disinfected seawater, right? So that means you're either going to autoclave it or you're going to give it some time next to a UV lamp and making sure that there's nutrients in it as well. And then taking a very small amount, we're talking microliters, of your raw culture from the seawater. You can add those into different areas and then just set it aside, have some of them, and then change the environment. So some of them brighter, some of them darker, some of them warmer, some of them colder. And what you'll find is that of the raw seawater sample, assume this is it, of that raw seawater sample, there might be 20 or 30 different organisms in there, right? And if you break it out and kind of separate it into, and I'm talking like 50 different tubes, and you're gonna, can you grab me a centrifuge tube before I write down there, Finn?
How I can separate zooplankton from phytoplankton? Because most of the time zooplankton and phytoplankton coming together in a net plankton. And I cannot separate zooplankton and phytoplankton from each other. And then after that, zooplankton eats all phytoplankton. And maybe after 2 hours, 3 hours, I don't have enough phytoplankton.
I hear you. One way to do that is to physically separate them by size. So you can screen them first. I'm assuming you use like a phytoplankton net to go gather them up first. Are you using like a trawl, a phytoplankton net?
Yes, same trawl. I threw it in the sea, and same trawl, I take it out.
Okay. So from that trawl, your sample is going to come in like this. One of the ways that what I recommend using, and it's funny that I have one right here, what you'll want to do at times is to, do you have access to glass pipetters?
Yes.
Okay.
Yes, micropipette.
Yep, you'll want to take your micropipette, and you're going to and you're going to want to draw it out really thin, and then you're going to break it, and you're going to take a piece of aquarium tubing. I know this is going to sound gross. Do you have a dissecting microscope you can use?
Yes, I have.
Okay, so get your dissecting microscope, put a piece of aquarium tube to the back of this, take the aquarium tubing and put it in your mouth. You're going to sit on the microscope. If you're like me, you have to take your glasses off. You're going to go here and you're basically going to use, it'll take about a day, but you'll get good at going through under the microscope, finding the cell you want. You'll pull it up into here. And then I use, I like to use these little tiny, matter of fact, I was doing this recently. I like to use these tiny Petri dishes.
Plates, Petri dishes, yes.
Take them, get them all laid out in front of you. Just number them. So all you have to do is throw whatever cell you want into it in there, and then separate those out. And then that's going to be what you're going to be growing out a little bit later on. You're going to bottleneck the culture if you only put one in. So let's say you can, if you have the acumen to go ahead and grab a bunch of them that are all the same, you can use that, right? Otherwise, one of them will be just fine as long as it's going to grow the right way. I like to get 2 or 3 at least. That way there's a little bit of genetic diversity if they do decide to sexually reproduce or at least exchange their genetic material at all. Did that make sense?
Yes, thank you very much. Thank you. I want to ask about a different class. Maybe you have another class, or do you have any archive about recording your video, how I can take it?
Yeah, so I've recorded this video this time. I had a couple of them, but I didn't record them. So it sounds like you're more of an advanced user, and we're more than happy to do that so that you have a more advanced questions and techniques that we can go and talk about. But I'm just now getting on to recording a lot more things and putting them online. But we do have a whole bunch of YouTube things that are on our website or on our YouTube site called The Dirty Lab Code. It's Algae Research and Supply. Because if your lab code is clean, you're not working. And if all your buttons match, you're not doing it right anyway, too.
Thank you very much. Thank you. I appreciate about. I like microalgae so much.
Awesome. Great. Cool. Any other questions?
Yeah, I've got a couple more. One, you were mentioning in the talk about using a carboy. So when I had just been reading around, I just heard about using aquariums. So I was wondering, for you, if you try growing it in a carboy, what would you have to keep in mind? Because I guess like the air circulation there is going to be different, and I don't know.
Yeah. So the important things to consider when you're talking about any kind of culturing is you need to think about light, you need to think about temperature, and you need to also think about mixing. We didn't talk about that in this class, but whether you're using an aquarium or carboy, you want to have some gentle mixing because the cell itself is going to be consuming materials right around it. So it's going to be actively pulling in nitrate and phosphate and other things. And it's going to deplete the area immediately around the cell. So it's kind of like a black hole consuming everything around it. So that mixing makes that boundary layer get sheared off so you can reintroduce nutrients into that boundary layer. So whether you're using an aquarium or carboy, it almost doesn't matter as long as you're able to meet the needs of the culture. Now, one of the reasons we like to use carboys is that you can more greatly enclose the entire system. Now, that's important if you're growing a microalgae that isn't, as we call it, weedy, like a weed. If it wants to grow, spirulina is a good example of this. Spirulina, if you keep it at pH 10, very few other organisms are even capable of growing there. And even if they do, spirulina puts out different molecules in the water that basically says you can't use that nutrient. It ties them up. They're called siderophores. And they tie up mainly metals like iron so that other organisms can't have access to them. They're ruthless. So
if the carboys can be a little bit more expensive, and if you get the glass ones, they're heavy as heck and dangerous. I like the plastic ones because I got kids in my lab running around sometimes. Yes, you can.
Do you have to worry about the algae, I'm not sure, or the spirulina or whatever, like growing onto the wall and like forming a film there that would block the light?
Yeah, so when we produce spirulina in a farm, every day we actually take literally a broom and we push broom the ground, or the bottom of the pond. So when you're growing spirulina, pretty much if you want it to grow very healthfully, you don't have to necessarily clean it, but you need to mix it. You need to get everything up off the bottom. And then every week or two, depending on how much of this dead spirulina settles down, you'll want to remove that. That way you can keep the living happy cells that are going to be suspended in the culture going. Otherwise, they're going to sink to the bottom and die, and you need to get rid of those. The reason is the stuff at the bottom is going to decompose, and that's going to change some of the chemistry of the water. It's actually going to, as that decomposition, it's respiration, so it's going to drive the pH down, which will invite a whole bunch of other organisms in there. And with spirulina, you want to keep the pH up because it's an extremophile, and that's where it works the best, with a high amount of alkalinity and a high pH. Alkalinity is different from being alkaline. Alkaline usually denotes a high pH, but alkalinity is the total amount of buffer. And spirulina is really good at handling that higher alkaline environment and higher pH as well.
Okay. And then another, I think, question would be, in the guide, you were saying that adding 1 milliliter of nutrients for each 10 grams of wet weight harvested. So I was wondering, in terms of a volume, since I guess I could buy a kitchen scale, but I don't have one. So I was wondering, like, would that be like 2 tablespoons, or how much, about what would 10 grams be, you know?
Oh, in wet weight, a gram of water is 1 milliliter. So okay. Spirulina wet is almost all water, even if it's just a paste. Okay. It might be a little bit, it might be 1.1 grams per milliliter. Just a little bit more dense than water itself. So you could assume 10 milliliters would be equal to 10 grams.
Okay.
Does that make sense?
Yep.
That's the metric system right there.
Yeah.
Another question. Sure. I have.
And I think we'll wrap it up now. I don't know if we're gonna. I'm surprised that it's let us go this long. But go ahead.
What is your common way and protocol for counting the microalgae? Is it a hemocytometer slide or a spectrophotometer? Oh, a section.
I like the Seshi stick. We made these. God, it took me a long time to find the right material. And
how I can make this? Is it possible I made?
Yeah, you should. It's just a millimeter ruler with a target. We've got these online, and we're happy to send one to you. I think they're $2. So, and you can get these wet. You can bleach them when you're done, so you can use them over and over again. They're pretty awesome.
Okay.
But you can also use we use a spectrophotometer. And if you do that, you either want to do it at the green range, so that's around 540. Otherwise, you want to do it at around 750 nanometers. That way you're outside of photosynthesis because it gets goofy with how they're absorbing light. You can also use a turbidiometer, but you want to make sure that you're going to, again, be outside of look at which wavelengths that manufacturer of the turbidity sensor absorbs. You can use a hemocytometer, and online I've taken the Seshi stick depth with a handful of different strains and done that to cell counts. So you can see cell count to Seshi stick depth. And then I also did dry weight. So those 3 factors are all online for spirulina and for Nannochloropsis, which are 2 of our main workhorse organisms.
Do you have any paper or guideline that we can use it?
For
culturing from the base culture of microalgae? Do you have any guideline or any paper, any things that
we have our algae culture manual that comes with every one of our orders. And inside there, we go over all sorts of different basic culturing techniques that you might come across. So general algae instructions are here on this side. That's on page 15. And then on 16, we go into some more stuff. And here we describe how to use the.
We can order from your website, yes?
Yeah, everything's available on our website as well.
Okay. Thank you very much.
Oh, you are very welcome.
Thank you.
All right,
This is really interesting.
Thanks so much. We appreciate you guys being here. We've got algaeresearchsupply.com. We've got all sorts of educational kits. Brainy Brinies are one where we feed the microalgae to brine shrimp, and you can quantify both of them there, as well as algae beads, which are a really great way to demonstrate photosynthesis in elementary or in the classroom. So, all right, guys. Well, thanks so much.
Thank you. Thank you.
And we're going to be trying to do another video tomorrow as well. So take care. Thanks so much for joining us.
Thank you. Take care.
Thank
Good
you.
day. Bye-bye. Bye.